Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. Today we are going to study uh, eukaryotic transcription. Transcription in eukaryotes. Okay, when we are talking about eukaryotic transcription, it means the process of transcription that occurs in eukaryotes. Well, we will take this topic from uh, a research paper, uh, transcription and translation, that is written by the authors. You may see them on screen. So previously we studied introduction to transcription, then we studied different components of transcription, then we studied prokaryotic transcription, and today we are going to study eukaryotic transcription. Although eukaryotic and prokaryotic transcription are basically same, although eukaryotic and prokaryotic transcription are basically same, eukaryotic transcription has some distinctive differences from prokaryotic transcriptions. Well, transcription means the formation of RNA transcript from DNA. Okay, a single stranded RNA copy is made from double stranded DNA. So the basic theme is same, but there are distinctive differences between the transcription processes occurring in two major domains. Eukaryotic transcription and prokaryotic transcription have distinctive differences. Eukaryotic transcription is more complicated. Well, the process we will study now, it will be more complicated as compared to transcription in prokaryotes. So eukaryotic transcription is more complicated. There are some reasons for this complexing. There are certain reasons. Number one, in eukaryotes, there are much more genes to be recognized and transcribed. When we come to eukaryotes, they have many chromosomes. For example, we study that in prokaryotes, there is only one chromosome present. Prokaryotes are haploid, they have only one chromosome. But when we come to eukaryotes, there are much more genes to be recognized and transcribed. Similarly, eukaryotes deal with these challenges with several ways. And because the process is complex, complicated, so eukaryotes deal with these challenges with several ways. The first of them is that in transcription, three types of RNA polymerase are involved. In eukaryotic transcription, there are three types of RNA polymerases that are involved. This is the first. But the first of them is that in transcription in eukaryotes, there are three types of RNA polymerases that are involved. Secondly, before RNA polymerase 2 enzyme begins RNA synthesis, it requires the binding of many proteins to the promoter. Okay, before RNA polymerase 2 enzyme begins RNA synthesis, it requires the binding of many proteins to the promoter. While some of these proteins, known as general transcription factors, bind DNA before the enzyme binds, some of them bind after enzyme binds. So there are certain proteins that bind to DNA before the enzyme binds, and there are some of them proteins. There are some proteins, some of them bind after the enzyme binds to DNA. The other reason of having complicated structure of eukaryotic transcription is that in eukaryotic cells there is nucleus. Another reason why this process is a complicated process is that in eukaryotic cells there is a distinctive nucleus. There is nucleus. Nucleus is present in eukaryotic cells. The nucleus is a main decisive uh, feature of eukaryotic cells. This nucleus is a main decisive feature of eukaryotic cells. Genetic material of the cells is stored in the nucleus. RNA synthesis takes place in the nucleus and required to be processed before transfer from nucleus to cytoplasm for translation. Genetic material of the cells is stored in the nucleus. Okay, RNA synthesis means transcription also takes place inside nucleus, and this RNA synthesized during transcription requires to be processed before transfer from nucleus to cytoplasm for translation. The final reason is that for transcription, the template genomic DNA is organized into chromatin in eukaryotics. When we come to eukaryotic organisms, the template genomic DNA is organized into chromatin in eukaryotic organisms. Some of chromatin structure can prevent the access to DNA. We know that there is some junk DNA that is never expressed, so some of chromatin structures can prevent the access to DNA. This feature of chromatin makes eukaryotic gene regulation a very complex mechanism. Okay, so when we come towards process, the first process is initiation of transcription, start of transcription. As mentioned before, as we have described before, in prokaryotes, transcription begins with the subunit of RNA polymerase. Holoenzyme recognizes -10 and -35 promoter regions of a gene. So when we talk about prokaryotes, in prokaryotes, transcription begins with the binding as mentioned before, in prokaryotes, transcription begins uh, with a subunit of RNA polymerase. Holoenzyme recognizes -10 and -35 promoter region of a gene. After the start subunit is decomposed and enzyme continues to synthesize RNA in a transcription bubble moving along the DNA. This is a process we have already studied in previous lecture in prokaryotic transcription. That transcription begins with the subunit of RNA polymerase holoenzyme that recognizes -10 and -35 promoter region of a gene. And after the start, the subunit is decomposed and enzyme continues to synthesize RNA in a transcription bubble moving along the DNA. Similarly, in eukaryotes, RNA polymerase II enzyme cannot recognize promoter sequence itself. In eukaryotes, RNA polymerase II enzyme cannot recognize promoter sequence itself. However, unlike bacteria in eukaryotes, polymerase is required to bind to the promoter region, which is an integral part of holoenzyme. So, unlike bacteria, when we talk about eukaryotes, polymerase is required to bind to the promoter region, which is an integral part of holoenzyme. General transcription factors do not take place travel with RNA polymerase during the RNA synthesis. The general transcription factors do not take place travel with RNA polymerase during the RNA synthesis. They recognize promoter sequences and bind it, and they have a duty to take RNA polymerase enzyme nucleus. They provide the right region to initiate transcription of enzymes. So they uh, have to provide the right region to initiate transcription of enzymes. Six general transcription factors have been designed as transcription factor 2A, TF2B, TF2D, TF2E, TF2F, and TF2H. RNA polymerase 2 is then separated from general transcription factor and initiates transcription. Although the detailed studies of this process are continuing, a protein tail of RNA polymerase 2 subunit called carboxyl tail domain (CTD) is interesting. Well, there are detailed studies that are going on that are continuing a protein tail of RNA polymerase 2 subunit called carboxyl tail domain is interesting. It is known that this section participates in many critical phases in the RNA synthesis and start phase in processing. It is known that this section that participates in many critical phases in the RNA synthesis and start phase in processing. This carboxyl tail domain is located near the region of the new RNA synthesized from the polymerase. So this carboxyl tail domain is located near the region of the new RNA synthesized from the polymerase. After the phosphorylation of carboxyl tail domain with one of the general transcription factors, the start phase ends up and the elongation phase begins. The start phase ends up and the elongation phase begins. Now we are going towards moving and studying elongation phase. Here, it is thought that the phosphorylation weakens connection of RNA polymerase 2 with other proteins of starting complex and allows elongation. So what happens here? Phosphorylation happens, and this phosphorylation weakens connection of RNA polymerase 2 with other proteins of starting complex, and now it allows elongation. Elongation. As described in prokaryotic RNA synthesis, eukaryotic elongation also takes place in transcription bubble. Eukaryotic elongation also takes place in transcription bubble. However, the newly formed RNA is different in prokaryotes and eukaryotes. The newly formed RNA is different in prokaryotes and eukaryotes. The carboxyl tail domain of the larger subunit of RNA polymerase 2 plays an important role in the process of elongation control. So there is CTD carboxyl tail domain of the large subunit of RNA polymerase 2 plays an important role in the process of elongation control. It was noticed that CTD phosphorylation occurs. It was noticed that CTD phosphorylation occurs with the efficient elongation transition. Efficient elongation transition. So it has been noticed that carboxyl tail domain phosphorylation occurs and it occurs with the efficient elongation transition. An intact CTD is required for elongation control process. CTD is formed by repeating a series of 7 amino acids many times. So there are 7 amino acids that are being repeated many times in a series, and this CTD is formed by repeating a series of 7 amino acids many times. These repeating units act as a binding site for some enzymes and some required proteins in coating, attaching, and cleaving of RNA. These repeating units of 7 amino acids act as binding site for series for some enzymes and some required proteins in coating, attaching, and cleaving of RNA. Because of being close to occurrence of new RNA from CTD polymerase, this region is an ideal place to control connecting and oscillation of proteins which are needed in the process of formation of new RNA transcription while RNA synthesis is continuing. So this is elongation phase. This is elongation phase of transcription. So the message on DNA is being read and RNA transcription is going on. So here we see that this region is close to occurrence of new RNA from CTD polymerase. This region is an ideal place to control connecting and oscillation of proteins which are needed in the process of formation of new RNA transcription while RNA synthesis is continuing. RNA elongation continues until the conserved sequences close to 3' are recognized by the enzymes. This RNA elongation continues until the conserved sequences close to the 3' these are the sequences AAU, AAA, AUU, are recognized by the enzyme. Then enzyme cuts the RNA about more than 20 bases from the 3' end. Then enzyme cuts the RNA about more than 20 bases from the 3' end. After cutting 150 to 200 adenine nucleotide portions, poly-A is added. So after cutting 150 to 200 adenine nucleotide portion, poly-A is added. Therefore, AAUAA sequence of protein coding genes is called as polyadenylation signal. And in last, we study termination. Although start and elongation stages have been well researched, initiation of transcription in eukaryotes has been well researched. Elongation has been well researched. Although start and elongation stages have been well researched, the mechanism of termination stage, especially for RNAP2 transcription, was unknown until recently. Termination is an important step for releasing RNAP from its own template. So termination is important step, and during this step, RNAP is released from its own template. Termination also prevents the formation of antisense RNAs. Termination also prevents the formation of antisense RNAs. Such RNAs may inhibit the formation of normal RNA. Such RNAs may inhibit the formation of normal RNA. So, thus, abnormal gene expression does not occur. The termination with RNA polymerase II does not happen in conserved region or at a fixed distance from 3' of mature RNA. Termination step in mammals occurs in anywhere from several base pairs to several kilobases downstream of 3' of mature RNA. And polymerase II termination can happen in different ways depending on the RNA 3' processing signal and termination factors which are located at the tip of the gene. So polymerase II termination can happen in different ways, and it depends on the RNA 3' that is processing signal and termination factors, and these are located at the tip of the gene. Two ways have been well studied, including poly-A independent and SEN1 dependent ways. So what is poly-A? Poly-A Poly-A dependent termination happens to many protein coding genes. Poly-A dependent termination happens to many protein coding genes where RNA polymerase II termination is engaged with RNA maturation event. Here, 3' of newly synthesized transcript is exposed to cleavage and polyadenylation. What happens here? 3' of newly synthesized transcript is exposed to cleavage and polyadenylation. This 3' processing reaction can be divided into two steps. This 3' processing reaction can be divided into two steps. Firstly, transcription of poly-A region follows the suspension of RNA polymerase II transcription and the endoribonucleolytic separation of newly synthesized transcript. So what happens firstly? First, transcription of poly-A region follows the suspension of RNA polymorph transcription, and it is co-related by endoribonucleolytic separation of newly synthesized transcript. Secondly, while upstream cleavage product is polyadenylated, Downstream cleavage product is decomposed. Upstream cleavage product is polyadenylated, while downstream cleavage product is decomposed. SAN1 dependent termination is an alternative termination mostly for non-coding RNAs. SAN1 dependent termination is an alternative termination, but it is mostly for non-coding RNAs. It is firstly discovered in Saccharomyces cerevisiae yeast. Okay, unlike messenger RNA, endoribonucleolytic division and or core exosome with exoribonucleolytic cutting at 3' of the yeast, snRNA and snORNA is created by a TRAMP complex, and they do not continue poly-A-tail in their mature form. Contain poly-A-tail in their mature form. So when we come about the second process, this is SAN1 dependent termination. It is an alternative termination mostly for non-coding RNAs. It is first discovered in Saccharomyces cerevisiae yeast, and unlike messenger RNA, endoribonucleolytic division and are core exosomes with exoribonucleolytic cutting at 3' of the yeast RNAs is created by TRAM complex, and they do not contain poly-A-tail in their mature form. Ribo RNA polymerase 1 termination includes less factors compared to RNA polymerase 2, but it is still a complex process. In yeast, the most important three major termination regions have been identified in the sequence of intergenetic spacer region. In yeast, the most important three major termination regions have been identified in the sequence of intergenetic spacer region IGS. In 90% of the yeast, RNA polymerase 1 is terminated in the first terminator. Cell box is the largest terminator in mammals. It is recognized by TTF1 termination factor for polymerase 1 for direct termination. In addition, in mammals, the formation of 3' of ribosomal RNA and oscillation of RNA polymerase 1 need a free factor. This factor called as PTRF polymerase 1 and transcription release factor is associated with RNAP1 and TTF1. Adding PTRF into in vitro assay is sufficient to release free transcript from whole DNA and RNA polymerase 1. So PTRF is added into vitro assay and it is sufficient to release free transcript from whole DNA and RNAP1. It can be removed. RNAP3 transcribes various short nuclear and cytoplasmic non-coding genes. 5S RNA, ribosomal RNA, U6, spliceosomal snRNA, tRNAs, and RNase P, RNA SE, MRP, adenovirus associated, and 7SK RNAs. RNAP3 is a very effective polymerase, not only because of their short transcription units, but also because the enzyme gets reinstalled quickly on similar transcription unit. So it is the advantage of RNA polymerase 3 that it is not only because of their short transcription units, but also because the enzyme gets reinstalled quickly on similar transcription unit. RNA polymerase 3 is able to efficiently terminate without an apparent need to further to other factors. RNA polymerase 2 has three classes. Each of their transcribed genes use a specific promoter. There are different classes you can see here, and they have a specific transcribed uh, genes that they use as a specific promoter. Although the use of different promoters, RNA polymerase 3 terminates transcription in a simple common consensus sequence of 3' of gene. In vitro transcription, the lack of specific subunits of RNA polymerase 3, these are the units, shows heterogeneous termination in accordance. The addition of C37, C53 heterodimer subunit is sufficient to allow recognition of terminator and to fix flaws of termination. So these are here to fix flaws of termination. It is suggested that C37 C53 complex or heterodimer subunit reduces the extension rate of RNA P3D. This reduction provides an increasing lag time for terminator elements to cause oscillation of transcript and RNA polymerase 3. C11 is an essential subunit that also mediates RNA cutting activity of RNA polymerase 3. And after this, there is comparison of prokaryotic and eukaryotic transcription that you can study by yourself. Uh, and Today we studied eukaryotic transcription. Tomorrow we will start translation. So till then, it's goodbye, Allah Hafiz, thank you.